Hello, students. Good afternoon. So, welcome you to the seventh week, to the tutorial session for the seventh week of the contents of the seventh week of this course, Introduction to Cell Biology. As always, I would also try to run you through and summarize first about the contents of this seventh week of this course. So, primarily, the in the seventh week of this course, the focus had been on the endomembrane compartments and the endomembrane components of the eukaryotic cell. So, as we all know, that the eukaryotic cell is a highly compartmentalized system, and it has, the plasma membrane. We have already studied about the plasma membrane, which is the outermost kind of which is which is the membrane because of which the identity of the cell is there, which makes the cell a completely different compartment and also allows maintaining the homeostasis of the intracellular environment despite changes in the extracellular environment, right? Now, even within the cell, the cellular biochemistry, all the cellular macromolecules are uh, divided, are kind of categorized and organized spatiotemporally in several membrane-bound compartments, right? Like the nucleus, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, peroxisomes, mitochondria, etc. So these intracellular membrane-bound compartments of the cell will together be referred, are together referred as the endomembrane compartments. And ultimately, there is a final plasma membrane or the cell membrane, which is the outermost membrane of the cell, um, which kind of makes the identity of the cell and is essential for maintaining the cellular homeostasis. Now, as we go forward gradually and all through, I think we will see one unifying scheme of event or one unifying mode of operation, that is these membrane-bound, intracellular membrane-bound compartments have, various different compartments have various different sets of proteins and biomacromolecules, and in many cases, various different environments, like for example, very acidic environments in case of the lysosomes, much neutral kind of environment, the cytosol, for example, mildly acidic environments in the Golgi, like which have a pH of around 6.5, where the lysosomes have pH of around 4.5 to 5.5, mostly around 5, whereas the cytosol will have a pH of around 7.4, and there will be different molecular crowding in different compartments. So in general, what I'm trying to say is that in these different intracellular membrane-bound compartments, you will say very different environments, very different sets of players, that is very different sets of biomacromolecules in terms of proteins, nucleic acids, etc. And they are like this because they are destined, or they're in the cell for performing a certain set of unique functions, right? But nothing also works in isolation. That is, there is also a crosstalk and interchange or exchange of biomacromolecules between these intracellular membrane-bound compartments. So we will try to look into these as well. So we will try to look into these individual compartments, as well as we will try to look at how these compartments, how there is a coordinated crosstalk and exchange of biomacromolecules in a very regulated and directional manner between these intracellular endomembrane compartments, as well as between these endomembrane compartments and the plasma membrane and the extracellular space, so to ensure the proper functioning of the cell. So to begin with, the three major compartments, at least two major compartments of this endomembrane system are the endoplasmic reticulum and the Golgi apparatus. So the endoplasmic reticulum, as we had also seen earlier, is continuous with the nuclear membrane, right? The nuclear membrane is a double membrane-bound structure, and the nuclear membrane runs continuous with the endoplasmic reticulum. And there is a different set of kind of stacked membranous structures which together form the Golgi apparatus. We will look into them individually first, and then we will also um, kind of try to see an overview of how these structures are formed and how these structures communicate among themselves and what are their functions in the context of the cellular physiology and cellular functioning. So this endoplasmic reticulum, the endoplasmic reticulum also consists of a kind of stack of membrane enclosed sheet-like structures which are stacked, which are also connected. I mean, there are not these are not four different membrane structures which are stacked and there is no membranous connection, but there are membranous connections between these endoplasmic reticulum membranous stacks as well, and they are stacked in a particular orientation. Now, as we will also look forward that the endoplasmic reticulum primarily serves as the hub for synthesis of so many different new proteins, right? Synthesis, so the ribosomes, so basically if we see the entire scheme of molecular biology, then there are, then your genes, that is your DNA, your genes are present, DNA is present within the nucleus in the form of the chromatin. Then there is transcription, you get the mRNAs within the nucleus. The mRNAs are then transported out of the nucleus to the cytosol or the cytoplasm, where the ribosomes, the translation machineries, they bind the mRNAs and they start synthesizing the new nascent polypeptide chains, right? Now, the polypeptide chain or the proteins, they are there are so many different proteins that are present in so many different endomembrane compartment of the cell. So how does a cell know that which protein needs to be that a particular protein, for example, is to be transported to which compartment? The answer is that all the proteins have certain unique sorting signals, certain unique amino acid signals, which are kind of the identities for the cell to know that this protein is to be destined to this particular cellular compartment. So there are several proteins which are called the secretory proteins, or which are present within vesicles or on the plasma membrane, right? So all of these proteins which are to be transported to these endomembrane compartments like endoplasmic reticulum, Golgi, and eventually to the plasma membrane, or even maybe they may be secretory proteins which need to be secreted to the extracellular space. They may be proteins which need to be sent to the late endosome, early endosome, uh, or lysosomes, peroxisomes in many cases. So are these proteins are synthesized by the ribosomes um, directly. Directly, we will see into this in details. This is synthesized by the ribosomes directly into the endoplasmic reticulum. So the ribosomes actually sit on the endoplasmic reticulum, and so the ribosomes bind the endoplasmic reticulum membrane, sit on the endoplasmic reticulum membrane, and directly synthesize the nascent polypeptide chain into the ER lumen, right? So, and then these nascent polypeptide chains are actually folded within the ER. There are several post-translational modifications like formation of cysteine disulfide bonds, or heavy glycosylation changes, or different other post-translational modifications that are necessary for the proper structure and function of the protein, as well as for the proper cellular compartmentalization of the protein, that is proper trafficking of the protein. So all these post-translational modifications and the folding of the protein is ensured within the endoplasmic reticulum, and that continues as the proteins actually move on from from this endoplasmic reticulum to the Golgi, from where eventually it is shipped into a certain direction or towards a certain destination of the cell. The endoplasmic reticulum. So the endoplasmic reticulum, as we will see, actually consists of two types called the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Rough endoplasmic reticulum is actually the endoplasmic reticulum to which the ribosomes are bound. right? So that is why they are called the rough endoplasmic reticulum. Smooth endoplasmic reticulum is basically the endoplasmic reticulum which does not have the ribosomes bound to them. That's why called smooth. However, one additional function of this endoplasmic reticulum, be it smooth endoplasmic reticulum or rough endoplasmic reticulum, is that the endoplasmic reticulum harbors several of these biosynthetic enzymes which are necessary for synthesis of the lipids that are integral components of the cellular membranes, right? Cellular endomembrane compartments as well as the cellular plasma membrane, right? For a cell which is, for example, you consider a daughter cell which is growing in size, you need extra membranes as well, right? Both for the endomembrane system as well as the plasma membrane system. So the source of and the integral component of this membranes are proteins as well as lipids. The membrane is a lipid bilayer, so the new lipids that are necessary for the cell are also synthesized within the endoplasmic reticulum, and they're incorporated into the endoplasmic reticulum membrane. And as we will see, then that this endoplasmic reticulum membrane actually serves as the membrane source for all the different endomembrane compartments as well as the cellular plasma membrane compartments in a regulated manner. Now, what happens is that from the endoplasmic reticulum, after the proteins are synthesized within the ER lumen and they are folded, and a certain extent of post-translational modification has happened, then these proteins are passed on. It's like a conveyor belt and need to be passed on to the next compartment of this endomembrane system, which is a Golgi apparatus. So from this endoplasmic reticulum membrane, what happens is several vesicles, right? Several vesicles bud out. Several vesicles are first formed. They bud out from the membrane, within which specifically the proteins that are to be transported to the next compartment are loaded. Then the vesicle pinches off, right? Then the vesicle is fissioned. It pinches off from the endoplasmic reticulum membrane, and these vesicles, now there will be several vesicles, right, which have um, uh, all kinds of different proteins, which are to be transported, which are to be delivered to the Golgi, right? So these vesicles are actually fuse together with the Golgi membrane structure. So the Golgi apparatus is also a membranous, is kind of a, several of these sheet-like membrane-bound sacs, which are called the cisterna. But their stacking is quite different from that of the endoplasmic reticulum, and the set of proteins the Golgi apparatus harbors is also quite different from the endoplasmic reticulum. And several of the other environments, as I mentioned, that the Golgi is mildly acidic, right? 6.5, having a pH of 6.5 to 6. Now, even within the Golgi, I'm describing the Golgi as a stack of several sheet-like membrane-bound sacs, but this is not so homogeneous. It is even within this stack of membranes, as you, which is located typically in the perinuclear region, that is um, just around the nucleus, just by the side of the nucleus. Uh, as you When you go from the nuclear side to the plasma membrane side, you gradually see a shift of the Golgi environment in terms of pH as well as the set of enzymes that are present in these compartments. And because of that, the Golgi cisterna is again further subclassified into three types. One, the Golgi cisterna, that is a Golgi membrane-bound sheet-like sacs, which are present towards the nucleus, are called the cis Golgi, right? Which receive these vesicles that have the proteins from the endoplasmic reticulum, and then at the face of the Golgi, or the membrane-bound sacs of the Golgi, which are located the farthest from the nucleus and closest towards the plasma membrane, are called the trans Golgi, right? So cis Golgi means a Golgi which is cis or close to the nucleus, and trans Golgi means a Golgi which is located the farthest from the nucleus, right? And in between the cis Golgi and trans Golgi, the membranous sacs that are present are called the medial Golgi. So the Golgi consists of the cis Golgi, medial Golgi, and trans Golgi in a direction um, which starts from near the nucleus and goes towards away from the nucleus, that is towards the plasma membrane, right? And these have been subclassified because of them harboring different sets of enzymes that bring about different post-translational modifications, different glycosylation modifications or folding modifications of the proteins that pass through these compartments, right? Uh, the trans-Golgi network also actually performs a very important function. The trans-Golgi network can be thought of as a railway junction, as a very big railway junction. So you can imagine that in a big railway junction, you kind of get traffic of trains from all the different parts of the country, and you get so many people who are willing to go to so many different parts of the country from the same station, right? So there you have to manage this traffic of people that is which are kind of comparable to the proteins which need to go, need to be transported to specific cellular subcompartments, and the trains are kind of like the vesicles, the different vesicles that will be budding out from this trans-Golgi membranous sacs. So specific proteins need to be boarded onto specific vesicles, right? Which will then which would then be carried by these motor proteins, be it kinesin, be it dynein, or be it myosin, on specific railway tracks to ensure that these vesicles having the respective protein cargoes, be it luminal proteins or be it membrane proteins, right? The membrane proteins, again, whether it is a transmembrane protein or GPI anchored protein anchored to certain leaflet of the membrane, whatever protein cargo the vesicle has is delivered to a suitable destination where it should, where it was destined to go, where it should go, right? So, so this is kind of the, in short, so, and as you can imagine that there are so many different cellular subcompartments where the proteins from the trans-Golgi need to be delivered. Like it can be delivered to the plasma membrane. It can be delivered to organelles like lysosomes and peroxisomes, right? So, so as I already mentioned, that the endoplasmic reticulum, which is continuous with the um, nuclear envelope or the double membrane bound nuclear envelope, is actually a stack of sheet-like membranes, and consists of, it can be of two different types, a smooth endoplasmic reticulum and the rough endoplasmic reticulum, depending on whether um, the ribosomes are bound to the endoplasmic reticulum or not. Smooth endoplasmic reticulums are primarily the major hubs of lipid biosynthesis as well as biosynthesis of several of these steroid hormones, many of the sex hormones like testosterone, estrogen, progesterone, etc. These are steroid hormones. So they are synthesized within this smooth endoplasmic reticulum. However, rough endoplasmic reticulum also has lipid synthesis, but rough endoplasmic reticulum has a more important function of you know, synthesis of it. It acts as the hub of co-translational synthesis of these proteins, which are destined to go to these endomembrane compartments like endoplasmic reticulum, Golgi, extracellular matrix, plasma membrane, lysosomes, endosomes, right? Late endosomes, autophagosomes, as we will see, etc. And so this, as I already mentioned, is a function of the smooth endoplasmic reticulum, um, and these are the function of the rough endoplasmic reticulum. And as I mentioned that even within the rough endoplasmic reticulum, you will have several proteins, several sets of different proteins. Like there will be chaperones, like HSP70s, calnexin, calreticulin. There will be proteins like these prolyl cis trans isomerases. There will be proteins which facilitate the formation of of the proteins within the endoplasmic reticulum, which will be necessary for the proper protein folding, for the proteins to assume the um, necessary structure that is necessary for their functions, as well as there will be a set of proteins which are basically glycosyl transferases that will cause glycosylation of these proteins, and this glycosylation also, and we have so many different types depending on which sugar residues are being attached to the proteins and at which position. And there is so much of addition and trimming of the which happens sequentially to these proteins to eventually achieve the needed or the necessary final glycosylation that is necessary for their protein-protein interaction, for their protein structure and function, be it catalysis or it be it protein-protein interactions. So all of these modifications start, a considerable extent of these post-translational modifications of the proteins also begin within the rough endoplasmic reticulum. And also, one additional thing is that the rough endoplasmic reticulum also acts as a very important compartment for the protein quality control. That is, since it is the hub, such an important hub of protein folding, if the proteins fail to fold, most often these misfolded proteins, which also have much higher propensity to aggregate, so they are sequestered. They typically are not allowed to get released from the endoplasmic reticulum. They are kind of trapped within the endoplasmic reticulum. They're given more rounds of chaperoning for allowing them to fold, but if not at all, then they are degraded. And there is a very unique response within the endoplasmic reticulum, which is called the unfolded protein response, which is an endoplasmic reticulum stress response, but a very important protein quality control checkpoint for preventing the misfolded proteins to get circulated in the different endomembrane compartments of the cell, and allowing and ensuring that they are degraded so that they do not create pathogenic conditions which arise due to protein aggregation and misfolding, and to ensure that only the folded and properly modified proteins are getting the access to get released from the endoplasmic reticulum into the next compartment, which is the cis Golgi. And so basically, from this endoplasmic reticulum, when vesicles come out having the suitable proteins that are to be delivered to the cis Golgi, what they essentially end up doing is that they provide both new membrane as well as new proteins to the Golgi apparatus. And when vesicles emerge out from the Golgi um, that are destined to the next compartments of the endomembrane system, what they essentially end up doing is again providing the new proteins in a compartment specific manner, but also provide new membranes to different endomembrane compartments or the plasma membrane whenever they're fusing to those destination compartments, right? So ER is providing you the new proteins as well as the membrane for the endomembrane compartments as well as the plasma membrane. Yeah, so the electron microscopic images, of the endoplasmic reticulum shows that the endoplasmic reticulum membranous sheets, endoplasmic reticulum membranous structures can be actually both sheets as well as tubules, but the endoplasmic reticulum, which is located just outside the nucleus, that is perinuclearly, is typically sheet-like membranous structures which are assembled in a very unique way. So you can see that like in between two stacks, these endoplasmic reticulum membranes are not discrete. They have clear membranous connections. And the membranes are continuous, but these membranes are arranged kind of like a spiral, which is similar to the several of the spiral parking lots, right? Which we see in shopping malls quite often. Yeah, and so from the face which is located, the face that will be located farthest from the nucleus will be closest to the cis Golgi, and from all of these sheets, actually these vesicles can bud off with the suitable respective proteins that are to be delivered from the endoplasmic reticulum to the Golgi apparatus. Yeah, this is a nice schematic which depicts the co-translational delivery of the nascent polypeptide chain by the ribosomes into the endoplasmic reticulum, um, lumen or the endoplasmic reticulum membrane, right? So do consider this fact that uh, proteins of the endomembrane system, right? Proteins of the endomembrane system or even at the plasma membrane, if you consider, and we have two types. Like for the endomembrane system, there can be proteins which are located, which are resident of the lumen of the membrane-bound organelle, or they can be membrane proteins, right? They can be membrane transmembrane proteins, or proteins that are bound to the single leaflet of the membrane. So in this co-translational mode of delivery of the nascent polypeptide chain, what happens is the ribosome binds the mRNA in the cytosol and starts the translation. But at the very beginning of the translation, when just a few amino acid residues of the nascent polypeptide chain has been synthesized, there is mostly in the in the N-terminal end of this nascent polypeptide chain, there is this unique set of 4 or 5 or 6 amino acid residues, which is called the signal peptide or the signal sequence. And for the proteins which need to be delivered to the endoplasmic reticulum, they will have a unique endoplasmic reticular signal sequence, which is recognized by a set of proteins, which is called, which are together called the signal recognition particle. So that will bind to this ribosome complex, this ribosomal complex, which has this nascent polypeptide chain, which has just come out, and then this entire assembly will sit on sit on the SRP receptor protein of the endoplasmic reticulum membrane, which is kind of a transmembrane channel protein, which will now allow the ribosome. So when the SRP is bound, the translation is halted temporarily. When this ribosome complex is actually, is installed on the this SRP receptor protein, which is the transmembrane channel protein of the ER membrane, then again translation resumes. And when the translation resumes, as the nascent polypeptide chain is further synthesized, more and more amino acid residues are being synthesized, it is actually being driven into the endoplasmic reticulum membrane. And gradually this signal peptide, whose function is already done, is actually cleaved by some specific peptidases which just recognize a signal peptide and cleaves it, right? Now the interesting thing is that while this co-translational translocation of this nascent polypeptide chain is happening, there are also other unique signatures of kind of clusters of amino acid sequences, which also ensure that whether the protein is a transmembrane protein and needs to be integrated into the endoplasmic reticulum membrane, or whether it's a lumen protein and thus it doesn't need any integration into the ER membrane and should be translocated entirely to the lumen of the ER membrane. So even if not, it's an ER protein. For example, if it's the protein which is a transmembrane protein of the plasma membrane, or transmembrane protein of the Golgi apparatus, or transmembrane protein of the lysosome, then also it will have the respective signal sequences so that it is integrated within the membrane at the level of the ER, right? And if it's a membrane protein, then it needs to integrate also in a particular orientation, right? Because, for example, it's an integral membrane protein which of the lysosome. So if its orientation is like, if it has a certain orientation, that is, for example, the face that needs to be within the lysosome, while incorporation within the ER will be within the ER lumen, right? So during this co-translational translocation, the proteins are also incorporated in a certain orientation. For example, if you have one plasma membrane protein, um, which has this kind of receptor domain that needs to face the extracellular matrix, right? Then you just backtrack its journey. It must have come from the Golgi apparatus within a vesicle, right? Now, when the vesicle fuses, it will deliver the protein to the plasma membrane. So within this vesicle, this globular receptor domain has to be within the membrane, right? Within this vesicle, basically. So if it has to be within this vesicle, then you can, if you backtrack even more, then the vesicle must have come from the endoplasmic reticulum. So on the endoplasmic reticulum, again, this globular domain must have been inside, right? If this orientation is wrong at the level of ER, if this globular domain is on the cytosolic end, then that's a catastrophe, right? So the orientation also matters a lot, and in many cases, mutations of the amino acid sequences leads to altered orientation, which can also create problems, right? So during this co-translational translocation, thus luminal proteins means proteins which will eventually be in the lumen of endomembrane compartments, like lysosomes, like the hydrolases of the lysosomes, or the proteins which are to be secreted out of the cell into the extracellular matrix, right? If in this vesicle you have one soluble luminal protein, then when the membrane fuses, this soluble luminal protein is released into the extracellular matrix. So these proteins are transferred into the lumen of the endoplasmic reticulum. Whereas if you have membrane proteins, membrane anchored proteins or transmembrane proteins, then they have additional amino acid sequences other than the signal sequence, which allows their incorporation into the membrane at the level of this ER in a particular orientation, right? Okay. Yeah. So one disclaimer in this regard is that typically in all the images, in the kind of the schematics that we saw before, the endoplasmic reticulum is kind of is illustrated in this manner, that it is located just outside the nuclear envelope, and then is the Golgi apparatus, and then is the plasma membrane. But within the cell, this is actually not the case. The endoplasmic reticulum is actually an elaborate network. It's almost half of the endoplasmic reticulum membrane contributes to around half of the total membrane bound compartments of the cell, including the plasma membrane. So you can and these confocal microscopic images, these kind of fluorescence microscopic images, um, very nicely show that the endoplasmic reticulum is actually distributed throughout the cell, whereas Golgi is primarily located in the perinuclear region, right? This Golgi membrane membrane bound cisterna, which are stacked together, are present primarily in the perinuclear region of the cell. There is a density of the endoplasmic reticulum in the perinuclear region, but the endoplasmic reticulum is present all over the cell, distributed all over the cell. And how do you see this? Is so basically these cells, both these cells are actually expressing a protein which is referred here as KDEL RFP. So RFP is actually a red fluorescent protein, but by molecular biology techniques, you have added a four amino acid sequence to this RFP, to the N terminal of this RFP. And those four amino acids are KDEL. KDEL is an amino acid sequence which is very characteristic of the ER resident proteins, and the cell has a very nice machinery of bringing these proteins back to the ER. So these proteins are always trapped and trapped specifically within the ER. Thus, by seeing the localization of RFP, which has this N-terminal ER localization sequence, you get to see actually um, the status of endoplasmic reticulum within the cell. On the other hand, there is this protein which is called GM130. GM130 is a cis Golgi protein. So it's a Golgi protein, but even within the Golgi, as we have already discussed, that there is heterogeneity, the cis Golgi, medial Golgi, and trans Golgi. GM130 is actually primarily the cis Golgi protein. And so for that, what we have done is we have used antibodies which specifically recognize GM130, and these antibodies are actually conjugated with fluorophores. So that's why on fluorescence microscopic imaging, um, when on fluorescence microscopic imaging, when you are seeing GM, the localization of GM130 within the cell, essentially you are seeing the cis Golgi within the cell. So these proteins are used as markers as unique protein markers for the different cellular compartments, because it has been established, it has been found that these proteins will be specifically found within certain unique subcellular compartments, right? The endoplasmic reticulum is distributed all throughout the cell. It's not just restricted perinuclearly, but the Golgi is primarily perinuclear, that is located just outside the nucleus, nuclear envelope. And as we have already mentioned, that the one that is closest to the nucleus will be called the cis Golgi, and as you go farther away from the nucleus towards the plasma membrane, it will be called the trans Golgi. So yeah, now let's look into the Golgi apparatus, right? So as we've already mentioned, the Golgi apparatus, just like the endoplasmic reticulum, is also a stack of membrane-bound structures, right? Membrane-bound, membrane-enclosed um, um, sacs, which are called cisternae, the Golgi cisternae, and but the stacking of the Golgi cisternae are quite different from that of the endoplasmic reticulum cisternae. We saw that the endoplasmic reticulum cisternae, they are the membranes, the different endoplasmic reticulum membranous sacs are not discontinuous, they are continuous, and they are organized in the manner of a spiral, which is kind of similar to the Parkin lattice type of arrangement. But the Golgi, in case of the Golgi apparatus, these Golgi membranes are discontinuous. Okay, there are discrete um, Golgi membrane-bound sacs or cisterna, which are stacked with one another, just like several rotis or chapatis or paratha stacked one upon another. And the one which is located close, the Golgi cisterna, which are located just outside the nucleus, just perinuclearly, will be called the cis Golgi, and then the medial Golgi, and then the trans Golgi as you move out from the nucleus to the plasma membrane. And the cis Golgi the cis Golgi actually receives the vesicles, the transport vesicles from the endoplasmic reticulum that carry both the proteins that are to be delivered to the Golgi as well as the new membrane that is to be delivered to the Golgi, right? And the trans Golgi, on the other hand, actually acts as an important. So the medial Golgi, cis Golgi, medial Golgi, and the early parts of the trans Golgi also play important role in both in the continuing the post-translational modification of the proteins, as well as the folding of the protein into their proper structures. Whereas the final trans Golgi network actually acts as the shipping side of the Golgi apparatus. It acts as the major sorting hub of the cell, where just like huge railway junction, from which different people have to be guided or trafficked to the different trains that they need to take to reach the different parts, the different destinations that they want to. Yeah, so this is basically, again, as we have already seen earlier. So on the right hand side, you see basically the blue is the nucleus, which is done with the DAPI. And perinuclearly, you see a three-dimensional reconstitution, a three-dimensional representation actually of the Golgi apparatus, which I think you can appreciate the stacked nature of the membranes of the Golgi apparatus. Yeah. So in the different, as I already mentioned, that the different sections of the Golgi, you get to see different post-translational modifications of the. It's like here you can see that in the as compared to the ER in cis Golgi, in medial Golgi, and trans Golgi. So in all of these compartments, you get to see some very different set of proteins, set of acetyltransferases and glucosyl transferases, as well as oligosaccharide or polysaccharide trimming enzymes, right? That eventually lead to addition of or the post-translational modification of protein to a very complex set of oligosaccharides and very unique set of oligosaccharides, which are necessary both for which function both like tags for the suitable transport and cellular subcompartmentalization of these proteins, as well as are necessary for the protein structure and function, right? Like catalytic activities or protein-protein interactions at the respective subcellular compartments. Okay. So now one important and interesting point to ponder and remember is that which we have also discussed earlier, is that the cytoskeletal network, especially in this case, the microtubule cytoskeletal components, uh, along with the motor proteins, that is the kinesin and dyneins, play a very important role, right? Not only in the transport, not only in the transport of vesicles, for example, in this case from the endoplasmic reticulum to the Golgi apparatus, or um, or in the transport of vesicles in between the different cisterna of the Golgi apparatus, right? From the endoplasmic reticulum to the Golgi apparatus, or in between the different cisterna of the Golgi apparatus, or in the transport of vesicles from the Golgi apparatus to the downstream subcellular compartments, like lysosomes, plasma membrane, peroxisomes, etc., late endosomes, autophagosomes, etc. Not only that, but these microtubule cytoskeletal elements also play important role in the spatiotemporal organization of the Golgi apparatus and the endoplasmic reticulum all throughout the cell, right? Because they also act as the anchor points that are necessary for the compact nature and stacked nature of these Golgi apparatus, along with the motor proteins which run on these microtubule tracts and are capable of interacting with the specific proteins of the Golgi membrane, right? Um, yeah, and this is there is a considerable amount of dynamics also in the organization of the Golgi apparatus, especially during cell division, right? Because in cell division, when the mother cell is getting divided into two daughter cells, then these endomembrane compartments, that is the endoplasmic reticulum and the Golgi apparatus, needs to be segregated as well between the two daughter cells. So there, what happens is during cell division, the Golgi apparatus and endoplasmic reticulum also gets fragmented and loses its. Usually, it stays in a very compact and stacked manner, but during cell division, there is considerable rearrangement of the Golgi organization, along with which is coupled with a considerable reorganization of the microtubule network. Um, so, which facilitates the segregation of the Golgi apparatus into two daughter molecules, two daughter cells, Golgi apparatus into daughter cells, and after the formation of these two daughter cells, that is after the cell cycle is cell division, the mitotic or meiotic cell division is done, then the Golgi apparatus actually comes back, collapses together, and forms this nicely stacked compact Golgi organization again, regains the compact Golgi organization, as well as several other things also influence the Golgi organization, especially like the cellular adhesion and cellular migration events, right? And while we saw earlier that the primary hub of microtubule polymerization and elongation kind of is the microtubule organizing center, right? The centriole, centrosome, and the microtubule organizing center, which is located perinuclearly. But recent studies also show that there is Golgi nucleated microtubules. There are Golgi nucleated microtubules. There are some microtubules are also nucleated at the Golgi apparatus, which also is important in the traffic and transport of vesicles in between the different Golgi cisterna, as well as in the transport of vesicles from the trans-Golgi network, for example, to various different subcellular endomembrane compartments, like the lysosome, late endosome, plasma membrane, etc. Yes, and so In case of the Golgi, like the proteins, they move. They move from the cis Golgi to the medial Golgi and the trans Golgi. Now this Golgi cisterna, the Golgi cisterna is cisternae are also very dynamic. That is, there is a Golgi maturation. That is, the proteins are continuously the protein cargos as well as the lipids. They're actually continuously moving from one Golgi cisterna to the next Golgi cisterna, right? And that's how they can move from the cis Golgi to the medial Golgi to the trans Golgi, so that they can see and encounter the respective enzymes and those environments for the proper folding as well as post-translational modification and their eventual delivery, that is eventual trafficking and sorting to the respective cellular compartment. Now, there are two models which are proposed for this, and it's not exactly clear that the mechanism of this is not exactly clear in the truest of the terms. But so these two models are that in one model, they say that there are vesicles that bud out, right? There are vesicles that bud out from each Golgi cisterna, that is from the cis Golgi vesicles bud out, which have the proteins and suitable lipids, um, and they fuse with the next cisterna, that is, for example, the medial cisterna. And that's how, and the same happens again from the medial cisterna, vesicles bud out with a suitable set of lipids and proteins that have a certain set of post-translational modifications and folding, that is, get the clearance to get loaded into the vesicles, and then they move to the next, that is the trans Golgi. And eventually, there is a vesicular budding out from the trans Golgi network, which are destined towards the final destination subcompartment, be it the plasmamembrane, be it the latent lysosomes, be it the um, lysosomes, etc. So that is one model of maturation. So this is kind of the anterograde model of maturation of the cisternal Golgi cisternae. Anterograde because you can see that this anterograde movement, and that is from the cis Golgi to medial Golgi to trans Golgi and then to the downstream subcellular compartment, is kind of in this direction, that is cis to trans and then to the subcellular compartment, which is mediated by vesicles that are moving in this direction. In the other direction, in the other model, what is being proposed is that the cis Golgi cisterna are continuously being formed. Cis Golgi cisterna are continuously being formed, and because of fusion of the vesicles that are derived or that are budding out from the endoplasmic reticulum. So that's how the new cis Golgi cisterna are always getting formed. And what happens is that there is this Golgi system are continuously moving from the perinuclear region towards the plasma membrane, and if some of these Golgi enzymes, which are very characteristic of this cis Golgi or medial Golgi or trans Golgi, so those are being brought back by some vesicles which are budding out from the trans Golgi, medial Golgi. So from the trans Golgi, for example, vesicles are budding out, which are bringing those enzymes which are very characteristic of the trans Golgi back to the medial Golgi, which are very characteristic of medial Golgi, but now this medial Golgi has moved forward to the plasma membrane and is destined to become the trans Golgi. So these enzymes, which are characteristic of medial Golgi, are coming back via this retrograde transport through these vesicles, and that's how this medial Golgi system are maintained. That's how the cis Golgi system are maintained, because they receive their enzymes from this medial Golgi and cis Golgi as well as from the endoplasmic reticulum. And from this trans Golgi, eventually it's getting fragmented into smaller cisterna, and finally into several vesicles are budding out from this trans Golgi network, which eventually lead to the delivery of the cargo, be it the protein cargo or the membrane cargo, into the respective subcellular compartment. Yeah, so this is kind of in gist the protein trafficking and protein sorting pathway that is operating within the cells, right? So especially the secretory proteins and the proteins which are destined towards the subcellular endomembrane compartments of the cell. So basically, the proteins are being synthesized by the ribosomes, and they're co-translationally translocated within the endoplasmic reticulum, which is located, which is, as we have seen, is distributed all throughout the cell. And from the endoplasmic reticulum, the vesicles bud out, which bring with them the set of suitable proteins and lipids that are to be delivered to the cis Golgi. And from the cis Golgi, there is cisternal maturation. So that can be, so the vesicles, that can be via vesicles, or that can be because of the maturation of the cisternae, from the cis Golgi to the medial Golgi to trans Golgi, where the proteins are now in this in the endoplasmic reticulum as well as within the Golgi. There is protein folding, formation of the protein disulfide bonds, and the protein post-translational modifications, which are necessary for the protein structure, protein function, protein-protein interactions, protein subcellular, further subcellular sorting or tagging of the protein, right? For example, mannose-6-phosphate is a very important tag that is recognized in the trans-Golgi network for the delivery of the proteins to the late endosome, right? So from the trans-Golgi, and also what happens is, for example, if some of these cis-Golgi proteins have been delivered, have by accident, some of these ER proteins, for example, by accident have moved to the cis-Golgi, or the some cis-Golgi proteins have moved to the trans-Golgi. Then these proteins are also brought back to the respective compartments via the retrograde transport, right? And that's where this sequence that we had already mentioned, this KDEL sequence, comes in very handy, because if a protein having KDEL sequence uh, is characteristically is strictly an endoplasmic reticulum resident protein, so if that protein has been transported by accident to the cis Golgi, then it is brought back by a very specific set of protein machineries, protein membrane, eventually cytoskeletal element and motor protein elements to the endoplasmic reticulum. Now, the trans-Golgi network acts as a major hub of protein sorting. So the vesicles formed from the trans-Golgi network, having suitable proteins, and they are then destined towards a particular subcellular destinations like the plasma membrane or the late endosome, lysosomes, early endosomes, etc. Now, let's look into one more endomembrane compartment of the cell, which is a very important compartment and player of the behind cellular physiology, which is the lysosome, right? So as we all know that the lysosome is the degradative bag of the cell. The lysosome is highly acidic. It has a pH of around 5. It has a plethora of several hydrolytic enzymes, plethora of acid hydrolases, which can be of so many different types, right? It can be nucleases, proteases, lipases, glycosidases, phosphatases, sulfatases, phospholipases, etc. And it has this ATP, it has this ATP pumps, ATP dependent H plus ATPases, these H plus pumps, which use ATP energy to drive H plus, pump H plus into the lysosomal lumen to make it so high, to make it highly acidic. And this high acidity is necessary because many of these hydrolases, nucleases, proteases, lipases, etc., which are present within the liposomes, are functional at their best, are best functional at the acidic pH, right? So because of that, maintaining this acidic pH within the lysosomes is necessary. Um, and However, consider this fact that lysosomes play very important roles in so many different cell types. In every cell type, lysosome plays a very important role. Um, in case of it acts as a major regulator of cellular signaling. So many of these cellular signaling proteins like EGFR, VEGF receptors. So for all of these receptors, when there is ligand which are present on the plasma membrane, After ligand binding, they generate a particular signal, a signal transduction cascade within the cell. But many of these proteins too cannot be allowed to constitutively keep on signaling, right? The signal cannot be constitutively on. So many of these proteins are endocytosed, and then the vesicles are transported towards the lysosome. Then these vesicles, which have these receptors, fuse with the lysosomes, and then within the lysosomes, these receptors are degraded to shut off the signaling, right? That is, the lysosomes play important role in protein recycling or turnover. In protein turnover, they play important role in the degradation of protein aggregates as well. Malfunction of lysosomal degradation of misfolded proteins can lead to accumulation of toxic aggregates, which in many cases cause neurodegenerative diseases or lysosomal storage diseases, right? And lysosomes also play an important role in cellular nutrition. For example, many of the cellular nutritional cargoes are quite large enough, so cells engulf them via endocytosis, or large enough so they engulf them by phagocytosis, or they can also engulf cargoes from the extracellular space via endocytosis. So that also leads to generation of endosomes or phagosomes within which the cargo is present. Complex uh, macromolecular cargo is present, right? Now these endosomes or phagosomes, if they are transported towards the lysosome via again these motor proteins which run on the microtubule tracks, and then there is fusion of these vesicles with the lysosomes, and then the lysosomal degradative enzymes, is all of these hydrolases come into picture. They digest and degrade these complex macromolecules into simpler macromolecules, simpler biologically utilizable macromolecules like amino acids, nucleotides, phosphates, and simple lipid molecules, which are then transported, there are several proteins of the lysosomal membrane called the permeases. They then allow these simpler biological macromolecules into the cellular cytosol and respective cellular compartments so that they can be reused by the cell for protein synthesis, lipid synthesis, and other cellular biochemical pathways, right? So the lysosome acts as a major source of providing the cell of degrading these complex macromolecules um, into simpler biological reusable molecules, which are then reused. That is, it acts as an anaplerotic kind of organelle, which supplies the simpler biological molecules, which act as can be used as raw materials for synthesis of these complex biological macromolecules, again important for protein turnover, as you can already imagine that for these macrophages and dendritic cells, um, the lysosomes act as very important components, because these lysosomes will be very necessary for degrading and killing of the pathogens that are being engulfed by these dendritic cells or macrophages. In that antigen processing, while the major histocompatibility complex, also lysosomes play an important role. Okay, lysosomal enzymes, and by the way, one mention-worthy thing in this respect is that the lysosomal membrane also, like these lysosomes, have so many lipases and so many proteases, right? But the lysosomal membrane, so how is the lysosomal membrane stable? I mean, these proteases, the lysosomal membrane also has several of these lysosomal transmembrane integral membrane proteins like LAMPs, lysosome associated membrane proteins, LAMP1, LAMP2, and these lysosomal membranes are also made up of lipids, right? But they are digested and hydrolyzed and cleaved by their own hydrolytic enzymes. In case of these lysosomal transmembrane proteins like LAMPs, they are heavily glycosylated. Again, these glycosylation, all post-translational modifications actually come from this endoplasmic reticulum and Golgi, right? And these post-translational modifications, in this case, the glycosylation of these lysosomal membrane proteins, actually protect them from being degraded by the lysosomal proteases. Yeah, so the lysosome also gets its proteins and lipid membranes from the the protein secretory pathway components, protein secretory pathway endomembrane components, that is the endoplasmic reticulum and the Golgi. From the Golgi apparatus, like within the proteins are being synthesized again by the ribosomes, co-translationally into the rough endoplasmic reticulum. It is then transported the cis Golgi, medial Golgi, trans Golgi, and all through this process, these proteins have their respective folding and post-translational modifications, very specific glycosylations, which act as tags for recognition in the trans-Golgi network for the delivery to the lysosomes, as well as eventually, as we see that within the lysosomes, provide them the resistance and protection from being degraded by their own hydrolysis and hydrolytic enzymes. And from this Golgi apparatus, eventually the vesicles bud out with the lysosomal enzymes, right? Lysosomal enzymes, that is lysosomal hydrolases, lysosomal membrane proteins, lysosomal V ATPases, that is these H plus ATPases, which will eventually pump the H plus actively within the lysosome to create the acidic pH. And these vesicles eventually, fuse to the late endosomes and lysosomes. And also nascent lysosomes come out as vesicles, which eventually gain these V ATPases and hydrolases and form the mature lysosomes. So that's how the lysosomes also get assembled by gaining their hydrolases, by gaining their membrane proteins, by gaining the V ATPases from this Golgi apparatus, endoplasmic reticulum Golgi um, trafficking pathway, even finally from the trans-Golgi network. And the lysosomes, the mature lysosomes, which have the hydrolytic enzymes, can fuse with the endocytosed vesicles, and fuse with the endocytosed vesicles that are harboring complex macromolecular food materials, or can actually fuse with the autophagic vesicles, which are formed within the cells, which actually have cellular compartments, some cellular organelle, for example, mitochondria, which is not functioning properly. That is autophagic vesicle, which has some dysfunctioning cellular compartment, right, which needs to be degraded. So that it's for two reasons, that it's malfunctioning can be prevented so that cellular physiology can be maintained. And the second thing is that if it is degraded, then its complex macromolecules will be degraded back into simpler biological molecules, which can be reused by the cell for the normal cellular biochemical activities, right? So in truest of senses, this fusion of lysosome with the autophagic vesicles, which engulf the dysfunctional cellular compartments, allows their turnover to prevent the dysfunction, to kind of clear the dysfunctional organelles, as well as to recycle them into simpler biological macromolecules which can be reused for cellular biochemistry. Autophagy is in 2016, Yoshinori Ozumi was awarded the Nobel Prize for autophagy. Autophagy, which in Greek means self-eating, is a very important pathway or response of the cell to the dysfunctional cellular components or subcellular membrane enclosed components, or for example, misfolded proteins and protein aggregates that accumulate in the cytosol. So what happens is basically, so if you have misfolded proteins which are aggregated in the cytosol, or aggregated within the endoplasmic reticulum, for example, or Golgi, or you have these dysfunctional organelles, for example, the mitochondria, then one membrane starts assembling around this, and eventually closes on itself to form the autophagosome, which is the membrane bound compartment that harbors these. It is also unique in its own sense that it's a multi-membrane bound compartment which harbors all these cargo, that is functional organelles, the protein aggregates, etc., which need to be degraded for the clearance, right? And for the clearance and turnover, that is recycling. So this membrane-bound vesicle, which harbors these cargoes which are to be degraded, self-cargoes, uh, should be reminded, self-cargoes which are to be targeted towards degradation. Now, this multi-membrane bound compartment is called the autophagosome. So this autophagosome then fuses with a mature lysosome, which harbors all these hydrolytic enzymes, to form a structure which is now called the autolysosome, where these cargoes which were to be digested now meets the lysosomal enzymes, the lysosomal acid degradative enzymes, hydrolytic enzymes. And after the degradation, the simpler biological molecules, for example, amino acids, nucleotides, etc., lipids, which are generated, are then transported out by these lysosomal permeases into the cytosol so that they can be reused again by the cellular machinery, right? In some cases, however, what happens is that there is also endocytosis of food vacuoles or something else. For example, there can be phagocytosis of bacterial cells, etc. These phagosomes or endosomes to form an amphisome and finally fuse with the mature lysosome and degrade them for the turnover. In case of the plants and early protists, instead of the lysosomes, they have a very similar compartment, but with much more number of functions, with much more varied functions and diverse functions, and also with some these membrane-bound sacs, which are much larger in size as compared to the animal cells lysosomes, which are called vacuoles, right? So the plant cells and the early protists, they have vacuoles, which are the organelles which are very similar to that of lysosomes, but perform more number of functions, are capable of performing more varied and diverse functions, and are much larger also in size as compared to the animal cell lysosomes. So, for example, they have several functions. They can be food vacuoles. They play an important role in storage of various ions and various food materials. Now, depending on the plant tissues, especially in case of the plant storage tissues, it can be contractile vacuoles in case of early protists, where they pump excess water out of the cell and help in maintaining the osmotic balance and also prevent cellular lysis um, in face of osmotic stress. And in mature plant cells, you also get to see very large these central vacuoles, which can also perform storage functions as well as important functions like these plant vacuoles have the capability of swelling up because of accumulation of more and more water, which also leads to the increasing of the plant cell size and the turgor pressure in case of the plant cells. They also store pigments. Metabolic byproducts are deposited, inorganic ions are deposited, as well as they can also store these defensive secondary metabolites of the plants, um, which the plants synthesize mostly for their defense, against herbivores, but often also have medicinal properties found to be important for human beings. Uh, and obviously, there are also these lytic vacuoles because they are like of acidic pH, which are equivalent to the lysosomes of the mammalian cells and animal cells. And these vacuoles have a very nice capability of storing more water in them in response to osmotic changes. And because of that, the lysosome, these vacuoles increase in size, also triggering the increase in plant cellular size, and therefore a controller of the target pressure of the plant cell, right? Several Nobel Prizes have also been awarded for this, for elucidating the different subcellular compartments, the endomembrane compartments. like the Golgi, the protein sorting machinery, which George Palade contributed in considerably. Albert Claude and Christian de Duve played an important role in elucidating, in finding out the organelle lysosome using subcellular fractionation methods. And Yoshinari Osimi was awarded the Nobel Prize for studying the important role of autophagy um, in 2016. So this brings us to the conclusion of today's tutorial session. I hope you were able to understand a few of the things. If you have any doubt, you can ask now. Okay, so if you don't have any more questions, then um, let's meet in the next class tomorrow. I hope you will be able to do this week's assignment. All the best for that. Bye.